In this video, I will review details we discussed regarding translation elongation in prokaryotes, and I will talk about translation termination. In another video, I will follow this up by talking about translation organelles such as mitochondria. We have previously covered how elongation requires an assembled ribosome ready to go on an mRNA with a start codon correctly positioned. And from that point, in order to enter into cycles of elongation, we need to have tRNAs that have been charged by their cognate aminoacyl tRNA synthase, or tRNA ligase, and we need elongation factors like EFTU, which will deliver the tRNA to the A site of the ribosome, and EFG, which is needed for translocation, movement of the ribosome from one codon to the next. It's important for all of these different factors to continually be recycled, to continually recharge tRNAs, or supply more energy to EFTU, which normally is associated with GTP, and also to add more GTP to EFG. This all means that for every step of elongation, we are using at least the equivalent of 3 ATP or GTP to recycle and recharge all of these factors. These steps, however, might not happen all at a uniform speed. We also previously discussed how the usage of different codons can influence the rate of elongation. Remember that the frequency that you see certain codons occurring in a open reading frame is thought to reflect real physical constraints and interactions. For example, codon usage, or the use of one synonymous codon over another, might be reflecting tRNA abundance. So in certain coding regions, you might find where you want to have very fast translation, you're going to be using codons for which the corresponding tRNA is present in very abundant or very high amounts, because the low amount of a particular tRNA would lead to a longer wait period for the ribosome on those codons. Codon pair bias is a little bit less clear, but it may represent certain unfavorable interactions between certain tRNAs when they occupy the A and P site. Again, a reflection or something to remember is that at any given moment you will have a ribosome which will have a peptidyl tRNA sitting in the P site and tRNAs sampling the A site. Regulation of the speed of elongation is important and has some consequences. For example, it could be useful for co-translational folding of proteins. Getting the ribosome to pause or slow down during certain parts of translating an open reading frame will allow certain parts of that protein to fold independent of things that will have not yet been translated. Elongation speed can also be important for gene regulation, particularly in prokaryotes. In these cases, proteins like NusG will actually help form physical links between a ribosome translating an mRNA and the RNA polymerase that is creating that very same mRNA. This often has the effect of helping to slow down the RNA polymerase so it does not outpace the ribosome. Normally, RNA polymerase is able to transcribe faster than the ribosome can translate, which is not surprising given the complexity that we've just seen in translation elongation. I'd like to invite you to challenge yourself with the following few questions. Why might this linking be important? What might happen if an RNA polymerase was outpacing the ribosome? And a more concrete example, what would happen if you used T7 RNA polymerase to transcribe a gene instead of the E. coli RNA polymerase? Go ahead and pause the video. Discuss this with your classmates online or any friends in person, or share a thought on the online forum. Here's yet another example of elongation, speed, and coupling between transcription and translation. This is actually a classic example of transcription and translation coupling. And this is the case of the leader sequence of the trip operon, the operon for biosynthesis of tryptophan. So keep that in mind. If this is an operon for biosynthesis of tryptophan, we're going to want to activate these genes when tryptophan is low. If the operator sequence is not bound by the repressor for this operon, then RNA polymerase will bind the promoter and begin transcription. It may transcribe the entire operon. It definitely will create an RNA that encodes the leader peptide, but after this point it might actually encounter a terminator. Here this region is also referred to as the attenuator. The leader peptide in itself has nothing to do with the rest of the function of the operon, but is important for regulation of the operon. In other words, the leader peptide doesn't help for tryptophan biosynthesis. It's not an enzyme. The function of this terminator, though, in conjunction with this leader peptide, is linked to translation. This attenuator sequence can adopt different conformations based on 4 different sequence regions. If region 3 and 4 are base pairing, they will form a terminator structure. However, an alternative pairing between regions 2 and 3 will prevent formation of the terminator, since region 3 will be paired with region 2 and won't be able to pair with region 4. Only the hairpin between regions 3 and 4 will form a terminator. I would encourage you to look at the sequence that follows region 4, the string of uracils, and think about why this is going to terminate transcription. A bit of a review from what we've talked about previously. In the leader peptide that precedes these regions, there are 2 codons which encode for tryptophan, UGG, UGG. If tryptophan is absent, there will be a lack of charged tRNA tryptophan, or the tRNAs that decode these codons, and the ribosome will stall when it gets to this point, waiting for that correct tRNA. Again, remember that it's not only that the tRNA has to be present, it needs to be present and charged. So if the amino acid is missing, there won't be any charged tRNAs to decode those codons. This stalling will promote formation of the base pairing between regions 2 and 3, or the non-terminating structure. And this will allow transcription to continue. Again, this makes sense. If tryptophan is absent, we want to transcribe all of these genes. However, if tryptophan is present, the ribosome will continue translating through this region. It will not be slowed down that much by the tandem trip codons. And by translating through region 2, this will encourage regions 3 and 4 to bind and form that terminator and terminate transcription. So in order for this system to work properly and to get this type of regulation, we need the speed of transcription to be coordinated with the speed of translation. And again, we see the speed of elongating ribosomes here, whether they're stalling at the trip codon or going through it efficiently, actually has an impact on the regulation of this gene. So in this previous example with the trip leader peptide, we saw that a ribosome will stall at a codon for which the cognate tRNA is either rare or absent. For the stop codons UAA, UAG, and UGA, there are no cognate tRNAs to decode these. So we would expect the ribosome to stall and sit at that point. But those codons will act as signals for different factors to terminate translation. What do we need to do to stop translation? Well, we know already one thing that we need to do is actually get the ribosome not to continue elongating, to stall at codons for which there are no cognate tRNAs. We are also going to want to release the protein that is bound to the P site tRNA, that peptidyl tRNA. We need to release the bond, break the bond and release that protein. And once releasing the protein, we also need to release all the other components, disassemble the ribosome. So we have, within termination, more than one activity we need to accomplish. Right? Simply breaking that bond between the tRNA and the protein is not necessarily going to cause the entire complex to fall apart. So for these activities, we will need two sets of factors. We have here release factors, which will specifically recognize stop codons. And these release factors will recognize these stop codons and promote the breaking of the bond between the protein and the P site tRNA. This will release the protein from the ribosome. This is going to require coordinated action of at least one of these recognition release factors, so RF1 or RF2, which have, even though they're proteins, will have certain parts of the molecule which can interact specifically with stop codons like UAA or UAG. Release factor 3 also helps promote this activity. You can notice that these release factors have a similar shape to other molecules which occupy the A site, like EFG or the EFTU GTP tRNA complex. We will also need to disassemble the ribosome, and for this we will need another set of factors known as ribosome recycling factor, or RRF. And this will disassemble the ribosome, dissociate the large and small subunits from each other so that they can be used again on another mRNA and thus recycled. Again, we can see that ribosome recycling factors tend to have a similar shape to other molecules which occupy the A site. These are all great examples of molecular mimicry. They resemble the shape of a tRNA and other proteins, allowing them to fit into that ribosome and allowing them to carry out similar yet somewhat distinct activities. For example, one of the domains on the release factor is able to decode a codon, but in this case, specifically stop codons. So when the ribosome is stalled at a stop codon, this factor can interact, but not when it's stalled elsewhere, like those trip codons we saw earlier in the video. Release factor also has a motif or a region that's highly conserved near where you would normally find an amino acid connected to a tRNA. And this GGQ motif is a hallmark of different release factors that's conserved among many of them. And this motif we'll actually see is responsible for the reaction that breaks the bond between the P site tRNA and the protein. To understand how this is accomplished, it's worth taking a little bit of a deeper look or closer look at what normally happens when we're adding an amino acid. During elongation, the amino acids on a cognate tRNA attack the acyl bond in the peptidyl tRNA. This is the reaction that occurs in the peptidyl transferase center of the ribosome, which is actually part of the large subunit, and this reaction is actually catalyzed mostly by interactions coming from the 23S rRNA. So in some sense, the ribosome is a ribozyme. It's an enzyme where the reaction is being catalyzed thanks to an RNA component. Again, you can see this here on the left side of the slide, where the A site tRNA with a single amino acid attached is in red. The N-terminal part of that amino acid will attack the C-terminal part of the protein shown in blue that's found in the P site of that peptidyl tRNA. When a release factor is present and successfully decodes a stop codon, the same type of reaction is going to take place, but the attacking group now is a warder that is coordinated or held in place by the glutamine that's found in the release factor, that glutamine which is part of that GGQ motif. This reaction is not going to add anything to that protein, but instead will allow it to be released from that tRNA. So, the release factor is going to act to release the protein. We will then have a ribosome which is devoid of protein with an uncharged tRNA in the P site, which is actually really the only time you're going to see that kind of a setup. Even when we assembled the ribosome in the first place, we had a charged tRNA, that initiator methionine tRNA in the P site. So here we have a ribosome which clearly has reached a stop codon. It has finished its job. It has released the protein, which can now fold and carry out its activity. Now ribosome recycling factor can come in and lead to the dissociation of these subunits, allowing them to initiate a round of translation elsewhere or on another mRNA. As a way of remembering the different events that occur in termination, think about what would happen if either one of these processes did not work correctly. If you did not have properly functioning release factors, you would not be able to release the protein. You would have invested a lot of time, energy, and amino acids to ultimately construct a peptide which is trapped, partially trapped in the ribosome. Likely, it will be unable to function. Without both release factor and then recycling factor, you'd be unable to dissociate ribosomes from this mRNA. You would end up having ribosomal subunits sequestered on an mRNA, and they would be unable to initiate new rounds of translation elsewhere. You actually would see protein synthesis grind to a halt, unless you continue to make new ribosomes. But of course, to do that, you need to translate ribosomal proteins. I hope you enjoyed this video on translation termination, translation elongation, and coupling. I will follow this up, as I said, with another video on translation as it occurs in organelles, in which case some of the details are different. Thanks for watching.